In this morning, speaker is Udo Seifert, and talk about current fluctuations in biomolecular systems.
Okay, thank you very much. And first of all, I'd also like to thank Abhishek and all the other organizers for inviting me. So it's always a pleasure to come to India and to discuss physics and non-equilibrium StatMech here. So I want to show you parts of our recent work, which I've put together under this title because it all deals with fluctuations, with current fluctuations on Markov chains motivated by biomolecular systems. So the plan is that I first recall the basic principles of stochastic thermodynamics, tell you a little bit about molecular motor, the F1 ATPAs we are looking at. And while investigating this system and comparing to experiments, we found something which we called tentatively fine-structured fluctuation theorem. So this is not meant in a mathematical sense, but it's just a kind of tentative notion for this phenomenon. And then towards the end, I want to discuss a new relation which we have found, I think, which is quite universal, and I hope that this will please the more mathematically or theoretically minded, because we have found it, but we cannot prove it. So there will be something left for the hardcore theorists in this audience. So the first part of the work I discuss is with Eva Zimmermann and Patrick Pizzonka, two grad students. This part is with André Barato, a very gifted postdoc who is now at the Max Planck in Dresden. Okay, showing very briefly this slide because it shows the philosophy of the stochastic thermodynamics. Classical thermodynamics, was developed 200 years ago for these kind of engines. And the question is, and the philosophy of stochastic thermodynamics is to explore whether the rules developed here can, and in which form they can, be applied to little molecular machines like this rotary motor, which is the F1 ATPAs. Now, most of you know stochastic thermodynamics is a very general framework which has emerged over the last 15 years or so. It's not a theory of everything. It applies to a large class of non-equilibrium systems which are characterized by the fact that non-equilibrium is caused by mechanical or chemical or hydrodynamical external forces. However, all these systems are always in a thermal bath which still provides a well-defined temperature. So all biology in our body takes place at 37 degrees. Still, it's a non-equilibrium system. However, temperature is well-defined, and that helps to develop this framework. And likewise, if we're dealing with chemical reactions like ATP hydrolysis, these molecules are provided at fixed concentrations, i.e. chemical potential is well-defined. And since we're dealing with small numbers of involved molecules, fluctuations are relevant. Typical examples is the unfolding of proteins, this machine I was talking about, but also thermoelectric transport. We did quite some work also with Keiji Saito, who is here, about the influence of a magnetic field in this kind of systems, but I'm not talking about that. So the basic idea is to take thermodynamic notions like the first law and the second law, entropy production, seriously along individual stochastic trajectories. And you'll find more general frameworks and also more references in this review article. Okay, so let me just remind you on the basic principle using this kind of paradigm, which is colloidal particles in time-dependent laser traps. So this is a optical tweezer, or you could imagine a colloidal particle driven across a periodic potential. So typically we're dealing with an overdamped situation where the velocity is given by the mobility times the total force. Now, the driving arises from the fact that we either have a non-conservative force like talked about yesterday, or that the potential or both are time-dependent through an external control parameter. There is the thermal noise coming from the aqueous environment, and we keep the strength of this noise given by the Einstein relation, i.e. related to the mobility. And then about almost 20 years ago, Ken Sekimoto pointed out that this Langevin equation can be cast in the form of a first law if these three crucial ingredients are identified properly. So for instance, the idea of applied work is that when you change the potential while the particle is sitting somewhere, this would be applied work. Non-conservative force times displacement is applied work. Internal energy in such a simple situation is just the position within the potential, and then the blue box works out if you identify the heat in this form, turns out to be the total force times the displacement. That's what you would have naively guessed anyways in an overdamped situation. But the key point is all these quantities are fluctuating along an individual Langevin trajectory. So heat is related to the entropy change of the surrounding medium, and therefore this is also a fluctuating contribution to the entropy production. And it turns out that on top of this heat, you can include a proper entropy for the system proper, which I call stochastic entropy. And then you can prove that with this definition of stochastic entropy or its increment, so log P is the solution of the Fokker-Planck equation, which corresponds to this Langevin equation. So when you integrate that entropy change of the system, and you add it to the entropy change of the medium, you get a total entropy change, and then you can easily prove in such a stochastic dynamics this integral fluctuation theorem, or then by Jensen, this kind of second law-like statement. Okay, so this is for Langevin systems, and I'm showing it here for 1 degree of freedom. Same holds for any number of coupled degrees of freedom. Yes, please. Well, a colloidal particle is in the micron size, and on the next slide you will see nanometer size. This would be my vision of a molecule of a protein. So it's small, nano, or micro. Is that okay?
The number of particles.
Oh, number of particles. Well, I mean, this is one particle. You could have n coupled particles, but of course you have the surrounding aqueous solution, which are many water molecules. They provide a thermal bath.
No, not the thermal bath, but the system itself.
One particle, two particle, n particles.
He means how many atoms in the polymer, how many degrees of freedom?
Well, I mean, for a colloidal particle like this, I have 3 degrees of freedom. If I join them to a polymer, it depends on the number of monomers which I have, but think of a finite number. I mean, this formalism is true for any finite number of coupled degrees of freedom. Okay, so since we want to deal with biosystems, we have to ask the same questions, first law, second law, for chemical reactions. So the idea here is this green box is an enzyme which has different internal states, and it can go from one state to another using a chemical reaction, like an ATP molecule becomes hydrolyzed, and then we use this kind of chemical master equation with rates omega nm and mn. The idea is each state here has a kind of intrinsic free energy, intrinsic internal energy, intrinsic entropy. And the key point is that you cannot choose these rates however you want. They have to obey thermodynamic consistency conditions. For instance, the log ratio of these rates has to be given by the free energy difference involved in this reaction, which would be the delta mu of the chemical reaction and the difference of the free energies of these states.
What is the superscript? The zero?
The zero here, this is the mass action law kinetics. So typically you assume that these reaction rates are proportional to the concentrations of A1 from going to left to right, and these square brackets are biochemist notation for concentrations.
So it's time-dependent rates?
No, they are not well, it depends. When the concentrations become time-dependent, then the rates become time-dependent. But you could also imagine that these molecules A1, A2, A3 come from chemiostats, which are kept at a fixed concentration, and then the chemical potential is fixed, and then the problem would have time-independent rates. And actually, what I'll be talking today is only about time-independent rates, stationary states. Okay, and then you can, similarly as before, you can write a first law where when such a reaction takes place, well, there is no work. that there's a change in internal energy, there's some dissipated heat, and then you just use these relations, work it out, and you get a relation between the heat and the chemical potential difference. Okay. Now, as I said, we're dealing today only with stationary states, and in stationary states there is a stronger statement about entropy production than this integral relation. This is the well-known fluctuation theorem, which was first discovered numerical work by Evans and co-workers, then shown for stochastic systems by Joel and Herbert and Jorge. Usually these are long-time statements, but if you accept this definition of stochastic entropy, you can easily show that it holds for finite times. And this aspect is important when you want to compare with experiments, because for colloidal systems, for biomolecular systems, it's hard to read this asymptotic regime. So I'm showing here data just as an illustration. This is the entropy production in this system. The histogram of the total entropy production, it's a little bit hard to see, but this red histogram is after 2 seconds. This is after 20 seconds. And you see these are highly non-trivial distributions. Still, these distributions have to obey this fluctuation theorem in finite time. Okay, now let me show you the molecular motor I'll be talking about for the next 10 minutes or so. So this is this rotary motor. This is typically a membrane-bound protein. You can isolate this part, put it on a cover slip, and if you now provide this molecule with an excess of ATP molecules, i.e. fuel, this purple subunit will turn around, just like a rotary machine. Now, this is a tiny molecule, so you would not be able to see that. So what you have to do is you have to attach a big latex bead or 2 of them to this gamma subunit, and then you can watch the motion, the rotary motion. Imagine you view it from the top side, the rotary motion of this, it's like a digit of a watch. And then if you plot the angle as a function of time, you get traces like this. So the blue curve is if you provide a little bit of fuel, you see individual steps. If you provide lots of fuel, this is the red curve, it's running faster and looks a little bit more continuous, but of course this is still fluctuating. Now when you see that and when you have in mind what I just told you, there arise natural questions like, how do you relate this kinetics to the thermodynamics? What about the first law? What about the efficiency of this as a machine? Suppose you apply an external torque, you can let run this machine against an external load, and then it acts like a motor, and this becomes a sensible question. However, for today, I want to focus on a different aspect, and before I do so, let me introduce the basic model. The key point is, and that has not been appreciated in the community before, you have to deal with the fact that you do not really see the motor. So you have to deal with both degrees of freedom. There is this big bead and there is the motor, and I'm now mapping this rotary motion to a linear motion just for simplicity. So there is this big bead. If you apply an external torque, it would mean an external force in a linear setting. And then this big bead is coupled elastically to the motor. And for the motor, I assume the simplest dynamics. I just assume individual steps. Each step will be 120 degrees in the rotary situation. Imagine a fixed step size in a linear situation. And each step would involve the hydrolysis of one ATP molecule. And that would drive the motor to the right. And it could then run and pull this bead even against an external load. So the dynamics is of a hybrid type. The bead obeys a Langevin dynamics, as I explained previously for the colloidal particle. The motor obeys a discrete dynamics, steps to the right, steps to the left. The ratio has to obey this local detailed balance condition. KBT is 1 in my units. So now the model is well defined. You cannot solve it exactly, even when you use a harmonic potential here, but you can easily simulate it. So it's 2 degrees of freedom, the discrete motor degree, the colloidal particle degree, and this is what you get in the simulation. Now we observe here in the experiment, we can see only the big bead. In the computer simulation, we can see the motor. The motor is discrete, these are the straight lines, and the fluctuating trace around it is the Langevin trajectory of the beat. So for instance, here it's a little bit hard to see because it's faint, but here, for instance, you see the motor steps, but the beat doesn't follow, and then the motor is pulled back by the beat. Here the motor steps, the beat follows. You see clearly these traces. You can look in the stationary state at the difference between motor position and beat position. So for very slow motion, you get a Gaussian. If the motor is faster, it's typically one step, or it can be one step ahead of the beat, and that's why you get this shoulder. And since this is a non-Gaussian distribution, you can obviously not solve it exactly. Okay, so I want to discuss this fluctuation theorem aspect, and I was motivated to start working on this molecule when I saw this paper some time ago by this Japanese group. So they looked at the probabilities distribution of the angle after fixed time. And they got data like this. When they plotted logarithmically the ratio p of plus delta theta minus delta theta, they got this almost straight line, which they interpreted in terms of the fluctuation theorem. So they used the simplest model, which would be just a Langevin equation for the angle at a constant torque, some noise, and then it's trivial to show that this probability to observe a certain angle increment after time t obeys this symmetry. The nice thing about this relation was that the slope in this simple model should be independent of the friction coefficient gamma, which means that you could infer the torque generated by this chemical reaction, by this motor, just by looking at the slope. That was the practical idea. However, there was a problem. If you look at these data experimentally and you plot them in this fashion, it turned out that the slope here became time dependent. The slope was decreasing with time. And they tentatively suggested that you should get this torque from the t to infinity limit of such a plot. Now, of course, this model is too simplistic. If this was the true model, there would be no time dependence. But as I said, you have to keep both degrees of freedom. And that's a typical effect of ignoring a slow degree of freedom. There is the motor and there is the beat. However, you see only the beat. Okay, so we looked at the same data in our model. These are the plots we get for this P of delta x, which corresponds very much to this data. This is our theory simulation results for the slope of this logarithmic representation. Indeed, it decays in time. However, it's rather easy to show that the delta t to 0 limit actually yields the average force and not the t to infinity limit. Now, before I talk about this t to infinity limit, I want to introduce a related system which we studied to explore that role of hidden slow degrees of freedom. This is with my colleague Clemens Bechinger in Stuttgart, where we looked at the following system. We took two colloidal particles, each driven across its own periodic potential, and when you view from top, you have this kind of geometry. The particles are paramagnetic, so in the presence of a magnetic field, they develop a moment, which then interacts, and thereby they interact via dipolar interaction. So it's clear that when you look at the combined system, it's a perfect non-equilibrium steady state which would fulfill all the theorems. So in particular, for instance, the total entropy production, which can be written in this form. So these are the respective velocities, and the state function u1 is the average conditioned velocity conditioned on the position. So it's easy to show that this corresponds to what I had on my introductory slide. That should obey the fluctuation theorem. Now, suppose you do not know that there are two rings. Suppose you only see this particle and you don't see the other one. Or suppose you do not even know that there is another slow degree of freedom coupled to this one. You could ask the question whether the apparent entropy production, which you would get by looking at the same expression but now marginalized over the unseen blue second particle, whether this quantity obeys the fluctuation theorem. Okay, this was the question we were asking here, and here are the data. So because the coupling is through a magnetic field, you can change that coupling. So for instance here, The black curve is what you get without coupling in this logarithmic representation. With coupling, you still get a straight line, but a slope which is less than 1. If you look at this slope as a function of the coupling strength, it decreases. If you look at the slope as a function of the time, it also decreases, as we have seen in this other example. Now, when I saw this data for the first time, I didn't really believe that there was always a straight slope. So I made them work hard, and we also did simulations. And it turns out you can create situations where this apparent entropy production in this fluctuation theorem type representation does not even show a unique slope, but rather some modulated curve here. And now I want to talk about this modulation aspect and what the slope means in such a situation where you have hidden degrees of freedom in more detail. And that's what we tentatively called fine structured large deviations. So I stick with this simple model where we have 2 degrees of freedom, the position of the bead and the position of the motor, and they are elastically coupled. So the total dynamics on the ensemble level is given by this kind of mixed Fokker-Planck master equation. So L1 is the Fokker-Planck operator for the bead, L2 is this discrete jump master equation type operator. Now we introduce the generating function with respect to the n variable, where n is the position of the motor. And in the long time limit, this assumes this large deviation form, but we will keep an amplitude in this form. So when you now go to a Legendre transformation, there is this rate function h of u, which is related to this alpha function here in the generating function. Okay, and now we calculate this rate function and we look at the data. So here, let me first show you the numerical plot. So again, here we look now at the distribution of the displacements after time t in this p of delta x, p of minus delta x representation. And the dots here are the data. So you see in this representation, you do not get a straight line, but you get a modulation of this straight line. In fact, it's easy to show that in the t to infinity limit, you have a kind of discrete symmetry. So the probability to observe delta x for the bead obeys this kind of symmetry when you move one step in this units given by the motor, which then directly implies that when you look at this ft representation, you get a straight line plus some antisymmetric, periodic antisymmetric function. Now, if you just look locally around 0, what you would typically do in an experiment, you would get a slope. If you had only a very small range, it would look like an apparent slope, but it turns out this slope is not determined by the entropy production. In order to infer the entropy production, you would have to take this discrete slope, which is given by the green line. This discrete slope here is given by this eigenvalue, and this eigenvalue just is half the entropy production in one step. So the message is, whenever you have these low degrees of freedom and you look at experimental data, if you use a too narrow a range, you might be misled by taking this slope for the entropy production. Yes, please. The periods are determined by the underlying spatial symmetry. The periods are determined by the underlying spatial symmetry. This function is given by these eigenfunctions, and therefore we have kept the amplitude in this large deviation representation. But there is no simple physical meaning to this slope here. That's the key message, because many people experimentally, they just take a slope and then they claim this is how we can infer the entropy production. This study shows that's not true if you have slow hidden degrees of freedom. Okay, and now this modulation actually is a very general phenomenon. So here is the rate function, And this variable is the, how do you call this, the empirical current, and V is the average current. So the rate function peaks at where this variable is 1. For this fluctuation theorem representation, you're always around 0. Now, if you zoom in and look at this variable, not on this scale, this is a scale which goes linearly in time, but if you look at a more fine-grained scale, you would actually see these modulations. And you do see them not only around 0, you would even see them if you looked carefully around the maximum. So we have the prediction that when you look at these fluctuations carefully in such a situation, you should always see these fine modulations. And the claim is, this holds not only for the simple model, whenever you have a model with spatial periodicity and hidden degrees of freedom, like suppose you had a more complex molecular model like this one, if you just observe the corresponding beat, you would see this modulation. Okay, now for this model, we also investigated the role of coarse graining in the following sense. I've emphasized, in principle, you should keep both degrees of freedom. But suppose you want to map that to an effective model where you only have 1 degree of freedom. This would be here on the right-hand side. So you imagine you only want to deal with the, let's call this still the motor, and you want to have coarse-grained rates, capital omega plus and minus, which contain the effect of having a beat. Now, of course, you have to get rules how to determine these coarse-grained rates, and the reasonable rule is that you want to get the same overall velocity in this coarse-grained model, and I also want to keep this local detailed balance condition. Now, for the simple case where the motor only has 1 state, these are 2 conditions for 2 unknowns, which you can easily solve. But the key point is that if you now compare this scheme, which I just described, to the usual model, there will be a crucial difference. In the usual model, what 95% of the people do is they ignore the beat and they apply the force directly to the motor in the exponent. And these factors theta minus and theta plus are the so-called load-sharing factors introduced by Kolomaisky and Fisher some 10 years ago. So this is what is usually done. That's how you get force-dependent rates for the effective for a motor particle. So usually probe is omitted. I just said this. And then it's assumed that the rates depend exponentially on the force. However, now having this full model and this kind of mapping, we can explore whether these coarse-grained rates are mono-exponential in the force. This is now as a function of the external force, and they are not. You clearly see different regimes here. The different colors refer to different sizes of this probe particle. So this is 1 micron, this is 4 nanometer, and you see only in the limit where the probe particle has vanishing radius, i.e. vanishing friction coefficient, you approach a straight line. This is what is usually done. And again, this is not specific to the simple model. We looked at Lipovsky's kinesin model with 6 different states, and there the effect is even more dramatic. In fact, we find that the stall force, that's the external force which brings the whole motor to a halt, in a full model depends on the size of the probe particle. This is now a prediction which could be tested experimentally. And as a last general statement for this model, this coarse graining is thermodynamically consistent because it actually leaves the entropy production invariant. So often in coarse graining schemes, you change the entropy production. In this scheme, where in the full model you would have a entropy production from the probe particle, this is the Fokker-Planck part, this is the master equation part, get a certain expression. When you look at this type of coarse graining, you can easily show that entropy production is conserved, which arises physically from the fact that the beat does not have cycles which burn free energy, i.e. which lead to entropy production. Okay, so much to this model. And in the last 10 minutes, I want to introduce you to this uncertainty relation. And I want to do this by inviting you to look at a model you all know in a different light. So let's look at an asymmetric random walk. Again, this could be a molecular motor stepping to the right, but allowing for steps to the left. And now I want to look at this from the perspective of a process which produces something. And this process produces steps. Okay, so on average, if it runs for a time t, the output will be x number of steps given by this expression. Now this output after certain time t is not sharp. After time t, let's say after 100 seconds, you may have sometimes went 50 steps, sometimes 52, sometimes 48. There is a variance in this process given by the diffusion constant. So I define as uncertainty the square root of variance divided by the output. Now from a biomolecular, biophysical, biochemical perspective, any such process requires free energy input. The molecular motor requires free energy input to run. There is a thermodynamic cost to this process, which is given by the current times the affinity times the time. What is the affinity? Well, the affinity here would be the delta mu of the ATP hydrolysis. Generally speaking, it's just a log ratio of these rates. Now you combine all these relations and you find, for this particular model, that the product of cost times uncertainty squared is given by this affinity-dependent function, which is bounded by 2 kBt. Yes, please.
I'm just trying to understand, why is this model different from a lazy random walk?
From a lazy random walk. I don't know what a lazy random walk is.
I mean, there's the probability of not doing anything.
Here, I mean, it's time continuous, so you jump with the rate k plus, and you go. It's time continuous. Sorry, I should have said this. It's time continuous. All my models are time continuous.
So the number of moves you make is
random.
That's always random. No, I'm confused. That's always random for a continuous time process. No?
Yes, it's always random. I mean.
So what's the difference? I'm just trying to.
There's no difference. This is a simple random walk. It's a simple asymmetric random walk where you step with probability k plus to the right in a time interval dt and with k minus to the left. It's what you know. Okay. I'm not telling you anything new. But I'm telling you that for this model, the product of cost times uncertainty squared is bounded. So if you want to have a small uncertainty, a small epsilon, then you will have a larger cost because you have to run it for a longer time. Okay, so
epsilon square is, which is usually called as randomness factor.
Yeah. Yes, please, Kiran.
This is irrespective of the definition of A.
Well, no. I mean, this is the definition of A because this is the thermodynamic definition. If you burn one ATP molecule, the affinity is given by the chemical potential of this, i.e. the concentration. So this you have to use to be thermodynamically consistent. Because if you don't use that, the thing will run even at equilibrium conditions, which you don't want. Now, key point is that when you now look at a more complicated process, so let's say we have a process where you have 6 states, still a unicycle, we can show that the relation we had previously still is true. So n is the number of states, A is the affinity of the whole cycle, and we can show that the product between cost and uncertainty squared is larger than this function, and this function is bounded by 2 kBt. Now you can saturate this bound, the first bound, by taking uniform, all plus rates the same and all minus rates the same. The second bound can be saturated close to equilibrium, i.e. in the linear response regime where you have the lowest cost but the longest time. The capital T is temperature. Little t. Little t is the time. A Kb is the Boltzmann constant. Well, temperature is 300 Kelvin if you talk about a system in the lab.
The rates don't depend on the time. Those are K plus.
Yeah, well, the rate, I mean, this affinity has an energy dimension. Chemical potential have an energy dimension. The rates are 1/2, but this is dimensionless. Henk, sorry.
I have an asymmetric random walk.
Yes.
What is temperature?
Well, if you have a molecular motor, the molecular motor runs in your lab, in your body at 300 Kelvin, and you have to take that seriously. You see, my message is you cannot use rates as you want when you talk about a system which is embedded in aqueous solution, where the aqueous solution has a given temperature. These are important constraints when you talk about biological systems. You can completely forget about that when you talk about ASAP, TASAP, but in a biological setting, you have to take this constraint seriously. And this constraint gives you additional information.
So what was
Yes, sorry, Joel?
what is a sigma?
Sigma is the entropy production rate, i.e. the rate with which you burn free energy. T is the temperature. So this is an energy per time. If I multiply with little t, which is the length of the duration of the process, I get an energy. Henk.
I'm a bit puzzled by the remark that you say the second inequality is satisfied if you choose the rates equal. If you set k plus equals k minus,
Yeah.
log is 0, so a is 0.
Yes. Well, I'm saying, you know, in the limit a to 0, this approaches to kBt.
But let's see, a over n is 0 times the coefficient of log.
Yeah, so it's the 1/a. This goes like 1/a, this goes like a. By L'Hopital, you reach the factor 2. Okay, so very briefly, how do we prove this, or what are we using? Well, you introduce this modified generator, then you have to look at this characteristic polynomial. Now, the key point is that the eigenvalue lambda, the largest eigenvalue, is a root of this characteristic polynomial, which allows you to determine the current and the diffusion coefficient just by looking at these coefficients. This is a method we learned from a little-known paper by Cosa, but I guess some in the audience would have guessed that. And then you can just throw this on Mathematica, calculate these coefficients, and do the numerics. So the surprise is that this holds even more general. You can take an arbitrary Markov network of states. So this would be a process where you have some internal states, and whenever you go from one gray box to the next, you produce a molecule or a step. And our claim is this inequality is correct for any such process. So any process which produces something, if you want this production to be sharp, i.e. to come with a little variance, i.e. to be of high precision, you have to invest a larger cost. So for instance, a precision of 1% will cost you at least 20,000 kBt. So we can prove that in linear response. We have done extensive numerics for different sorts of networks, but, and that's the challenge to the theoretically minded, we have not, despite trying hard, we have not been able to come with a rigorous proof of this inequality. We understand it on physical terms because we already know where it can be saturated. So in a sense, the best process are the unicyclic ones where you have uniform forward rates and uniform backward rates. If you add additional cycles to a unicycle, you basically increase the fluctuations without gaining much. That's my physical understanding, why it's also true in this multicyclic case. But I stress we don't have a mathematical proof. Now, it should be obvious that you can use this now that this has implications for many questions. Let me just show you one. You can now use that to infer reaction schemes of enzymatic processes just by looking at the fluctuations of the product formation. So suppose you have an enzyme, E, which can bind a substrate and then transform the substrate into a product and release it. So this top triangle would be what is usually called Michaelis-Menten kinetics. But suppose this enzyme could also bind two substrates, transform one to the product, transform the other one to the product, you would have a more complicated cycle. So as a side result of what we have done, you get a bound on this Fano factor, which is the variance divided by the mean. So our result implies that this Fano factor is bounded by this function. A, again, is the affinity, i.e. what you spend in one of these little cycles, and M max is the largest value of a quantity, which is the number of states in the cycle divided by the number of products. So for instance, in this cycle, you have 3 states and 1 product, so this would be 3. However, in the blue dashed cycle, you have 5 states and only 1 product, so this would be 5. So here we have randomly generated rates in such a network at constant affinity and plotted this Fano factor. These are 100,000 different rates. That's how we do the numerics. And you see all of these dots lie above this red line, which is this function. So now by measuring the Fano factor and by knowing the affinity, you can get a bound on this mx value, i.e. you can use that as a diagnostic tool to explore the network topology of unknown enzymatic schemes. So this is just one example what you can do with this kind of new inequality. And this brings me to my final slide. I've discussed the stochastic thermodynamics framework and this fine-structured fluctuation theorem coarse graining, and I've shown you how this dynamic uncertainty relation provides constraints on various quantities. And I want to stress here in front of this community that the mathematical proof for the multicyclic case is still open. Thank you very much.
Any question or comment?
I have a very naive question.
Yeah.
About your first molecular motor.
Yeah.
If hidden degrees of freedom are ignored, I mean, your message is that if hidden degrees of freedom are ignored, then you get erroneous results, right?
Yes.
But isn't that problematic because every experiment, ignores certain degrees of freedom, either out of sheer ignorance or coarse graining, whatever.
Yes.
So then why are things valid at all? I mean,
well, things are valid at all because you have timescale separation. You should not ignore hidden slow degrees. Hidden fast degrees don't matter. They equilibrate, they belong to the bar. So the key point is if you have hidden slow degrees which have a timescale which is like the timescale of the quantities you can observe, then the general message is the exact relations will be spoiled. If you have, that's a theory. Right? If you have timescale separation, it's fine. If not, then you're in trouble.
You mentioned at some point that the fine structure holds for any model with a spatial periodicity and these hidden degrees of freedom.
Yes.
Do you really need spatial periodicity?
It
seems to me that this fine structure should be, not in this particular shape, but it should be even more generally true. Don't you think so?
It might be more generally true, but I understand it in this case where I have the spatial periodicity. But of course, if the particles run in a periodic potential, then again you would have it. And if you don't have spatial periodicity, everything is flat. You just have, coupled Brownian motion. Then of course, I think you would not get it.
Right. But if you have, let's say, some more random.
Oh yeah, I don't know what happens in a random energy landscape. No idea.
With respect to the actual model you want to simulate. To model how much the overdamped, why the overdamped dynamic is so effective with respect to the damped one. You could have used the damped.
Okay, so I was showing the general framework for overdamped dynamics. You can easily do all the stochastic thermodynamics for underdamped ones, and our chairman has done interesting work on the role of when you keep the momenta and they have this different symmetry under time reversal. But basically, for these colloidal particles, experimentally, you do not get in the regime, or you hardly get into the regime where you would see inertia effects. And likewise for these molecular motors, I mean, this discrete dynamics is intrinsically, in a sense, overdamped when you have this Markov dynamics. So, but I think this is a very... Yeah, viscosity is enormous. Viscosity is enormous. So from a practical perspective, this is the regime you should look at. But conceptually, you can include the momenta. You have, in some aspects, you have to be a little bit more careful. But in principle, that's understood how that works.
If hidden variable is also slow, then if you integral out, then usually you're going to get a non-Markovian.
Exactly.
So
and
that's the non-Markovian effect.
Yes, and that's the reason why these exact relations which are proven for Markovian dynamics are spoiled. Exactly, yes. Effective dynamics is no longer Markovian.
Any other question?
I had a question which goes back to what Stefano asked before.
Yes.
So you were describing this very simple random walker. So current and variance is something purely stochastic.
Yes.
And then you bring the affinity from somewhere else. From thermodynamics, from physics.
Yes.
Can you explain how in general you can define affinity for any stochastic process? Or
well, for any stochastic process where you tell me the rule, you tell me the source of the non-equilibrium. If you have unbalanced chemical reaction, if you provide ATP at high concentration, then it drives, Le Chatelier, it drives towards burning the ATP. And when I burn one ATP and I know the concentrations, I know the delta mu, and this is the affinity. And I've choose my rates such that the logarithmic ratio obeys this given affinity, i.e. chemical potential difference. If I don't do that, this thing will run even if I have equilibrium chemical potentials, and that violates the second law. So whenever you spoil this relation, you violate this relation, you violate the second law.
Yeah, but you agree that KBT here is just a scale factor? You could
yes, I agree.
One.
Yeah, I'm not insisting on the KBT. I'm insisting on the blue stuff.
Yeah. So the blue stuff, can you define it in a general process?
Well, I mean, you can you see, if you take your favorite process with given rates, you can always define an affinity, right? But I mean,
it will be true, your identity in that case.
The relation, yes, it's true. It's true. It's true.
For any stochastic process.
For any stochastic process, we have looked recently at one of these ASAP models. Yes, it's true. But you have to have a process where the back rate exists, because otherwise it's trivially observed, because one side is infinity in these models. But yes, it's true. It's a mathematical statement. The KBT is not relevant.
That's what I wanted to.
Yeah, sorry. No, I was sorry for not being clear. It's a mathematical statement true for any network. Affinities are defined by these logarithmic ratios. Where is it? Here it is. Right? This is how you define the affinity for given rates.
Okay, and KBT is irrelevant.
KBT is irrelevant. KBT is irrelevant.
Okay. Thanks to the speaker again.